Hi, this is Jennifer King. I'm the Director of Science and Research for Lung Cancer Alliance, and today I'm going to talk about molecular testing and targeted therapy in the lung cancer space. So these are my disclosures. I do sit on advisory boards and receive speaking honoraria from a number of companies in this space. All of the funds are paid directly to Lung Cancer Alliance. Our agenda today is to talk, as I said, about molecular testing and targeted therapy, but what I'm going to do is go backwards. And I think this will make more sense as we go through the presentation. Normally, you would think about diagnosis of a lung cancer and then molecular testing and then the treatments. But for the purposes of today's talk, I think that really understanding the treatment space and where we are is going to underscore the importance of molecular testing. So I'll be starting with the new treatments. We're going to go through the evolution of how some of the older targeted therapies have really changed over time, as well as some new targets and a quick mention at the end on small cell lung cancer. So targeted therapy in general is different from standard chemotherapy because the drugs are directed at a specific DNA or protein change that's found in the cancer, as opposed to regular chemotherapy, which targets all very fast-growing cells. Target therapies are often given orally, and they also have different side effect profiles than chemotherapy or standard therapies, and the side effect profiles can be specific to the target that the drug is aimed at. So in non-small cell lung cancer, there are a number of approved targets, and I'm going to go a little bit through each one of these different targets. But the main ones that have FDA-approved therapies are EGFR, ALK, ROS1, BRAF, and just this week, NTRK, which had a new FDA approval. There's also a number of biomarkers for immunotherapies, but those are going to be in the following talk by Dr. Liu, who's focusing on immunotherapy for lung cancer. So I'm going to start with EGFR. And not so long ago, EGFR was the first target that had approved targeted therapies in lung cancer. Originally, these therapies were approved for metastatic lung cancer, and at first they were approved without any molecular testing. As we came to understand the science around these therapies, erlotinib and gefitinib, it became clear that these drugs worked on patients who had cancers that had mutations in the EGFR gene. So now a test is required that demonstrates mutations in the EGFR gene to get these therapies. The science followed this, and people started taking gefitinib or erlotinib, but after a while, they began to develop resistance to the drugs. So the drugs were effective for a while, and then the cancer started coming back and growing again. And this is work from back in 2005, but what was discovered was that in a lot of these cancers, there became another mutation in the EGFR gene that was in T790M. And you can see here from this study that a number of different T790M mutations were identified in different cancers, and this became the most common resistance mechanism to these true first-generation EGFR inhibitors. So the science led to more drug approvals, and as of just last year at this conference, what we saw was that there were now multiple generations of drugs targeting EGFR mutations. So the most common EGFR mutations in lung cancer are L858R, or an deletion in a part of the gene called exon 19. There were then, as of last year, 3 different drugs approved as first-line therapies if you had these mutations in your cancer: erlotinib, gefitinib, and afatinib. If, after being on those therapies, you developed an EGFR T790M mutation, then there was an even newer drug called osimertinib that was approved to treat the cancer. And that's where we were just a year ago. But what's incredible about the lung cancer field right now is the pace of research that's happening and how much we learn in just a few short months from all the studies that are ongoing. So since last year, we've had new drugs that are moving earlier in the treatment paradigm. We're seeing brand new drugs on the market, as well as new resistance mechanisms starting to develop. So since last year at this time, we've seen the results of the FLORA trial, which is first-line osimertinib, which was that newer generation drug originally used for T790M mutations, compared to one of the standard EGFR TKIs of erlotinib or gefitinib. And what you can see in the blue lines, which are osimertinib, is that there's an improvement in survival when osimertinib is the first-line therapy. This is true both in patients who have CNS metastases as well as those without. And this is really notable because those first-line, the original first-generation drugs, erlotinib and gefitinib, were not as good at crossing the blood-brain barrier as osimertinib. And osimertinib is much more effective in those who have CNS metastases. So this is an overall survival difference that's very significant. You can see this was published in New England Journal of Medicine just this year. And because of this, osimertinib is moving into the standard of care as a first-line option for patients who have EGFR mutations. In addition, we've seen other new drugs approved. This data was shown at the 2018 ASCO annual meeting by Dr. Tony Mock about dacomitinib. Dacomitinib also showed an improvement when used first-line against gefitinib. There were some additional toxicities, but dacomitinib was still US FDA approved on September 27th, 2018, giving a new choice for oncologists and their patients for what therapy they take first-line. In addition, we're learning more about drug resistance. So the FLORA trial I just showed you about osimertinib, there were patients on that trial who were able to give blood samples when they progressed on or discontinued taking osimertinib. And the blood samples from those patients, we were able to look at the gene changes by next generation sequencing and found the things that are potentially making these patients resistant to osimertinib. So common things were MET amplifications, a new mutation in EGFR called C797S, PIK3A mutations, KRAS, and BRAF. And so what this does is it gives researchers an idea of what mutations the cancers may develop that will make patients resistant to osimertinib, and then that helps determine what new second-line therapies or what potential combinations could be used for the second line after osimertinib. There's also been data shown on another EGFR mutation called G724S, which is actually sensitive to some of those second-generation inhibitors that we already have drugs for. So there's lots more coming. Despite all this work that's been done this year, we're going to see even more combinations, things that follow these EGFR inhibitors, and smart ways to take them and to sequence them going forward. In addition, and particularly since this is a screening audience and they were interested in early stage cancer, we're starting to see these EGFR targeted therapies move into the space of earlier stage cancer. So this is the SELECT study that was published very recently, and it was on 1A to 3A EGFR mutated lung cancer. Here they looked at the disease-free survival at 2 years compared to a historical control and found that there was in fact an improvement. And you can see the level of improvement compared 88% overall, which was different by stage compared to the historical. And in this case, erlotinib rechallenge could also lead to durable benefit. So if people stopped erlotinib, their cancer started to grow, and then they started it again, they could still see a benefit. So more work is needed in this space, but it implies that we may see some of these targeted therapies moving into early stage cancer where patients may have surgery or some type of treatment and then be able to take EGFR targeted therapies as an adjuvant or extra therapy afterwards. So all these advances I just showed you were specific to EGFR mutated cancers, but the story is very similar for some of the other mutations. For ALK, for example, which is often changed by gene fusion, there was an initial approval of CRIZOTINIB in August 2011 as the first ALK-targeted agent. Ceritinib followed shortly thereafter. But now what we've seen is that the ALEX trial, which was published in 2017, compared a newer generation ALK inhibitor called ALECTINIB to CRIZOTINIB first line. And so this was a particularly notable trial. You can see the ALECTINIB in blue. There is a very significant difference in progression-free survival when you took ALECTINIB first line compared to crizotinib first line. And here, again, we see a big difference in CNS progression. So ALECTINIB really is working much, much better in the central nervous system in the brain than crizotinib is. And that also translates to an improvement in overall survival for these patients. So ALECTINIB has also moved to, similar to the story in EGFR, with osimertinib moving to the first line, we're seeing ALECTINIB, this newer ALK agent, also move to the first line because of its efficacy and its improvement in overall survival. Importantly, we've also seen another ALK inhibitor this year, brigatinib, in the ALTA trial, ALTA first-line trial, show the same sort of difference where it is also performing better than crizotinib in the first line, and it also penetrates the blood-brain barrier better than some of the agents that were out there. So these stories are not just happening in EGFR. For ALK, we had those original 2 drugs. Now we have 2 drugs with better CNS penetration. We've already seen alectinib move to the first line. And in fact, just recently, there was another approval of a 3rd generation drug called lorlatinib that was just approved this month. So the ALK story is also growing, where we're getting more and more agents, and we're better able to select patients and start to try to understand which patients should take which drug when. This was a study that was done a while back in Cell Lines, but it compares those 5 ALK inhibitors across the top that I just told you about that are FDA approved against different type of fusions in ALK. And you can see that different drugs are better against different fusion partners. So the ALK field may become even more personalized, where it's not just that you have an ALK fusion, so you go on an ALK inhibitor. People are actually potentially going to be able to target which inhibitors might be better for you depending on what type of mutations that you have. So this is really exciting. We have these great drugs for EGFR. We have these great drugs for ALK. Where's this space going? What we're seeing is more and more gene mutations enter the fold as being targetable and having FDA-approved agents. So not only is it more than just EGFR, it's more than just EGFR and ALK now in non-small cell lung cancer. We've also seen crizotinib approved for patients who have cancers with ROS1 changes, and there's a number of other ROS1 targeted agents in clinical trials. In addition, if you have a BRAF V600E mutation, there's a combination targeted therapy called dabrafenib-trametinib. Dabrafenib and trametinib, there's 2 different drugs that are given in combination for patients with that mutation. And just recently, we saw an FDA approval for a new drug called larotrectinib that is actually targeting a whole new class of agents with those who have changes in the NTRK gene. This drug has a really particularly interesting approval because it was approved in advanced solid tumors for both patients who are pediatric, who were 12 and over, as well as adults. But you can see, and this is a figure from the paper that was published this year in New England Journal of Medicine, that there's patients with lung cancer who are included in this study. So NTRK mutations can happen in a percentage of lung cancers as well as a number of other types of cancers. And this drug, larotrectinib, has been very effective against any cancer that has a mutation in the NTRK 1, 2, or 3 genes. So we're just slowly adding to the arsenal of all of the different targeted therapies that are out there and the different gene changes that can be targeted. And this is great news for patients and the treating oncologists because it's giving more and more hope for different options that patients can go on. So there's even more coming. That's the excitement. You know, I'm looking forward to talking again next year on this topic because we've seen this year strong data on new RET inhibitors in non-small cell lung cancer in early phase clinical trials. There's a few new options there, and there's many other targets that are coming. So even if a patient doesn't have a targetable mutation with an approved drug, there may be a really relevant clinical trial for that patient. And it's a really exciting time in lung cancer. As I mentioned, clinical trials, they're so important. That's what's moving this field forward. We really encourage patients and their oncologists to look at lung cancer clinical trials, particularly with some of these mutations that might have a targeted agent on the pipeline. It really could be their best chance at care. This is something Lung Cancer Alliance is very passionate about, making sure that patients get on appropriate clinical trials, and we do have clinical trial navigators that can help with this process if needed. I also want to just give a brief mention to small cell lung cancer. Everything I've talked about so far has been in non-small cell lung cancer, but there are researchers looking at targets and targeted therapy in small cell lung cancer too. One example has been the target DLL3 and a drug named Rovati. Unfortunately, this drug, which had some promising early phase studies, this year did not do well in a later phase study, and that study, which a lot of us really wanted to be successful because it could have been the first targeted therapy to be approved in small cell, really did not show any benefit to the patients. However, this drug is still in trials in a different setting, in a phase 1, in a first-line maintenance type setting, and we're also seeing new drugs that are coming online that have the same target, because this one had some safety concerns. And so hopefully in a different setting, or some of these new drugs may alleviate those concerns. But this research is happening in small cell, and we're hopeful that we will get there, that we also have an approved targeted agent, and eventually many approved targeted agents for the small cell population as well. So as I mentioned, we're going backwards. So I just talked all about the new treatments and the evolutions of targeted therapies. I think now you can potentially understand how important testing is going to be. So we just talked about 5 different genes that have the FDA approved therapies, and many more coming in clinical trials. Patients are not going to be able to get onto those trials to get the appropriate targeted therapy unless they have molecular testing. So the NCCN guidelines currently say that, testing should be conducted as part of a broad molecular profiling. This means that you shouldn't be testing just EGFR or just ALK. You should be looking at a broad panel. This will help find those 5 mutations that I talked about, as well as potentially other mutations where there may be an appropriate clinical trial. And to really illustrate, I took the concept of this slide from a pathologist I saw talk recently, but to really illustrate why this panel is so important, if you think about doing multiple single gene tests on a patient, the picture at the top is a paraffin block that's used for tissue biopsies. You can see the size of the block. It's not very big. The biopsy tissue often is similar to the size of a pencil lead refill, and that would be embedded into that little bit of paraffin wax up there. And then for every single gene companion diagnostic, for every single gene test, you might use about 5 unstained slides. So you'd cut 5 slides off the top of that block. Now, if you're going to do an EGFR test and see if it's positive or negative, and then move to an ALK test, and then move to a ROS test, a BRAF test, an NTRK test, and then a test for a clinical trial, you can see how fast you will use up that block and not have any tissue left. So doing all these companion diagnostic tests one by one by one just doesn't make sense in this era of broad molecular profiling and being able to look at multiple different gene changes at one time. So there are FDA-approved panel companion diagnostics being done by next generation sequencing. There's 2 that have been officially FDA approved in lung cancer. The first was the OncoMind-Dx target test, which had 23 genes. It was approved specifically for non-small cell lung cancer for multiple different therapeutic indications, multiple gene changes that can point to those targeted therapies that I just talked to you about. And then a little bit later in 2017, there was an FDA approval for Foundation 1 CDX, which covers 324 genes and has this is for multiple different cancer types, not just lung cancer, and across multiple therapeutic indications again. So these tests do exist. They are FDA approved. One of the major questions is, how do they get paid for? How can my patient access them? Luckily this year, there was a decision by CMS, the Centers for Medicare and Medicaid Services, saying that CMS will cover these next generation sequencing tests that are FDA approved, if the patient has a stage 3 or stage 4 lung cancer, really a stage 3 or stage 4 any cancer, but in our case lung cancer, if they have not previously had this test, so they will only cover one test, but they will cover one of these advanced panel tests, and assuming that the patient is going to seek further treatment. And these are for the tests that have been FDA approved or that are all covered. So those ones that I just talked about are covered under this Medicare decision. Now, just because Medicare will cover it does not necessarily mean that private insurance will cover it, but a lot of private insurance do tend to follow suit. So hopefully we will see more broad coverage of these next generation sequencing panel tests so that patients can really find out what their best targeted therapy options might be. Importantly, though, we are seeing broad disparities in testing and potentially issues with guideline adherence. I showed you the current NCCN guidelines for panel testing. At the time that this paper was done that I'm going to show you, the NCCN recommended testing for 7 different mutations. But when this group looked at community practices, this was done mostly in the New Jersey area, what they saw was that patients were really way under-tested for not only all 7 NCCN recommended mutations where very few patients were getting the large panel, but even for EGFR and ALK, which had FDA-approved therapies at the time. So there was definitely under-testing happening. We know that this can be an issue in the community, and we're really urging that patients advocate for themselves, talk to their healthcare providers, and really try to make sure that they're getting broad panel testings whenever possible. So there are a lot of challenges in community-based practices. This was from ASCO of this year, of different issues that these practices may have in test selection, timing, tissue collection, and tissue availability is a huge problem, actionability, patient attitudes, and financial concerns. So challenges absolutely remain, but we're still trying to advocate for testing in a large scale way in a broad panel. So I will do a shout out to LCA here, where Lung Cancer Alliance has a program called Lung Match that's a personalized treatment and trial navigation program. Within this program, we can get patients molecular testing. There's a biomarker testing component that uses an IRB approved registry protocol. Patients get access to both genomic and proteomic testing. One of the nice things about this program is patients stay with their current healthcare provider, the patient consents, but then the healthcare provider is asked to sign the order. So we go out to the original treating oncologist to sign the order, and both the patient and the oncologist receive the results. The results are a combined report of both NGS as well as immunohistochemistry, biomarker testing. The reports have treatment recommendations, and they've been reviewed by an expert tumor board. And then the Lung Cancer Alliance treatment and trial navigators can call the patient and educate them on the results, what they mean, what they should be discussing with their doctor, and provide our free educational materials while explaining choices. And they also follow up with these patients to see if they have any other support needs. So this is a program that we are trying to get out there to both patients and healthcare providers to let you know that you can take advantage of this program. And it's generally at little to no cost, typically no cost to patients. So I would be remiss if I didn't also talk about liquid biopsy. Obviously, I've mentioned that tissue is a problem. Their tissue utilization, as I mentioned on that slide with the companion diagnostics, is an issue. Many lung cancer biopsies do not have a lot of tissue, and often there's not enough tissue for broad panel testing. So liquid biopsy can now be considered an option for patients who don't have a valuable tissue from a primary biopsy. Liquid biopsy is most appropriate for patients who have metastatic disease, where the disease is spreading and shedding cells into the blood. But in these patients, you can draw blood and actually see the DNA from the cancer in the blood. So patients who don't have tissue, who can't undergo a rebiopsy or won't undergo a rebiopsy, all could be great candidates for liquid biopsy to do testing. And there's even starting to be arguments for whether liquid biopsy should happen in parallel to tissue, because it may have a faster turnaround, it may have added value. And I point you to this recent JAMA Oncology paper about the implications of plasma-based genotyping. In this paper, patients who were found to have mutations by liquid biopsy, you can see some of the ones I talked about, EGFR mutations, ALK fusions, BRAF mutations, even MET, which we talked about is an up-and-coming clinical trial. Many of these patients had mutations that were found in the blood, and when they were found in the blood, and they were targeted appropriately, you could see a large change in the target lesion. So patients are responding to the drugs that are aimed at these targets. This allows many of those patients who didn't have adequate tissue to still go on an appropriate targeted therapy. In some cases here, there wasn't enough tissue and just the liquid biopsy was done, or just the liquid biopsy was done for various patient or physician reasons, and these patients were still able to go on targeted therapy. So plasma-based genotyping liquid biopsy is coming and is another option for patients. So our take-home for today are quite a few. When there is hope for patients who are diagnosed, as I think you will have seen, research is changing really quickly. There's a lot happening, and there's many new options and different treatment paths, even compared to just a year or two ago. So no patient should be told, oh, you have lung cancer, there's nothing we can do. There's a lot that could potentially be done, and patients really need to advocate for themselves, and healthcare professionals need to make sure that they're getting proper broad panel biomarker testing, because that's critical for proper treatment. New data is changing our treatment paradigm every few months, which is very exciting. Because of that, I want to give a shout out to some LCA unpublished data that patients actually want to hear about clinical trials from their treatment team. So you as screening professionals may be the first person that they're encountering when they come in about their lung cancer, or at that point, potential lung cancer. And so you are in a position to tell them that there is hope, that there are new treatments, and that clinical trials are often a great option for them to consider. So in summary, the screening work from those who are here at the screening conference, combined with advances in treatment, is going to make a huge difference in improved survivorship and quality of life for those who are living with or at risk for lung cancer. And thank you for all that you do.